Today I'll do a quick introduction to insectphilo.org and quick analysis. Now the idea behind this website and the data provided by it is a framework for community ecology, DNA barcodes and metabarcodes. So to demonstrate the general idea, I've obtained some published data in a regional DNA barcode study of some flies. These are some chironomids, and obtain the data from European Nucleotide Archive, align the data, and I just renamed them queries. So we have about 36 query colons from chironomid flies. So you could build a phylogeny of just your queries, which I've done here. And in community ecology, you'd often calculate, so you go beyond, say, OTU richness, and you can calculate phylogenetic diversity from this, gene tree, effectively. Now, the problem with doing that is, besides these internal nodes won't be well supported, but because of the very restricted regional sample, the branch lengths are going to be in some parts grossly underestimated, and you can see this in this figure. These red dots denote, this is for a different study, but the same thing has been done. These red dots denote some queries, but these have been placed onto a comprehensive reference tree, and when this is done, you get a much more accurate phylogeny, but also branch lengths are going to be a bit more reflective of actual distinctiveness between your OTU. So two aspects to improving the accuracy of your calculation of phylogeny diversity indices. So along these lines, this insect philo provides the reference data. It provides a phylogeny, and it provides a reference DNA barcode alignment. So we have these queries from some chironomid flies. So if we were to do this, then we go to insect phylogeny, and we need a reference phylogeny of Diptera. Currently version 1, and we have down here is a printout, but we need the NUBIC, which is a machine readable version of the phylogeny, synthesis phylogeny Diptera, and CO1 faster alignment. So save both of these and just move these into our working directory. So in a simplified analysis, what we want to do is align, so we have our queries and we have our reference dipter colon alignment. So we want to be doing profile alignment, which is a version of multiple sequence alignment where you have a fixed reference alignment. And in this quick demonstration, I'll use Clustable Omega to do this. There's various software for doing reference alignment, but this should work fine. Clustable Omega, we provide a profile alignment, or the reference alignment, which you've just downloaded from insect Philo. We have our queries for our phylogenetic community ecology study, and we just give a out file name. And we have references and queries in the same file and aligned. You can see the queries, I've just named these accordingly. So 38 queries, and then the rest of the file is all several thousand diptera co1s, and each of these faster entries corresponds precisely to a terminal label of the reference phylogeny. So in total 3,126. And next we just need to do a tree search. We use XML to do this. XML version 8 need to convert to Phylip format. And let's look at the command line settings, -s. We give the alignment. So again, this is references and queries. This is the output file. This is a default DNA model. A couple other things. Now importantly, we need to use the -g switch, which gives a constraint tree. Now if you don't do this, XML will just be inferring a phylogeny for everything, but we have a reference tree which a great deal of work and information content went into defining the topology of. So we need to set this with a -g switch, and we have a single outgroup for Mechoptera. So the reference tree used several thousand barcodes. Also, it integrated topologies grafted from analysis of over 100 published source trees, and also a transcriptome derived backbone topology. Now in running XML, there's more than one way this could be done. You could opt for the evolutionary placement if you have a lot of queries, but here there's not too many, so I'll just do a regular tree search and should not take too long. So several minutes to place about 36 geonomed queries onto a reference phylogeny of about 3,000 diptera terminals. And lastly, let's take a look. using any tree viewer. This one is our Archaeopteryx. Just search for query. And you can see they have been placed within the genus Diomesa and within some other chironomids. So after placement of your queries onto the large reference phylogeny, you can then go ahead and calculate phylogenetic diversity indices, and it should give you a lot more accurate indices, certainly for overall measures like uncorrected face PD should be a lot more accurate. And okay, so ends the quick introduction to insect phylum.